Ignazio Avella. He did his PhD in the institute where Maggie now is, the University of Porto in Portugal. But now he is, as we heard earlier, in Germany, the Justus-Liebig University in Giessen in Germany. His publication record makes it unambiguous that he's a herpetologist with a particular passion for snakes. And in recent years, his focus has been mostly vipers and their venoms. And that, I believe, Ignazio, is what you will talk about today. So welcome to EUVEN. The podium is yours.
Thank you very much, Ron. Very nice presentation again. And yes, I will just try to share my screen. Let's see if it should be all right. Okay, I think we're good to go. So yeah, thank you, Ron. As you rightfully said, I'm going to present like the results of a study again on venom, but this time we're moving from old world vipers, which have been my main focus until now pretty much, to crotalids, so pit vipers. And yes, short-nosed viper venom. Let's go. First of all, sorry for maybe sounding a bit cocky, but I figured that a bit of an introduction about myself would have been in place. So, who am I? As Ronald said, I'm currently a postdoc at the Justus-Liebig University of Giessen here in Germany. I mainly focus on the study of snake venoms, and as a side hustle, let's say, I am also interested in venom delivery systems. Definitely, as you can see from the pictures, I'm more into, I started from a herpetology background, and then eventually moved towards toxinology, which is now the main focus of my research. And I really try to analyze snake venom composition and variation and functional effects from an ecological and evolutionary framework. So yeah, there has been quite a bit of record, to be fair, about the analysis of snake venom variation at different levels from my side, both compositional and also like from the physical properties. Currently, I'm trying to developing some works on the venom delivery systems of cobras, which is also another project I'm quite interested in. But definitely the main focus is still on the venomics side. I'm a member of the Ludeca Lab, also known as Animal Venomics Group. And as you can see, like from the little silhouettes that I that I put here on this on this picture, many people work on arthropods, but the snake group is going strong, and we've been growing quite a bit over the last year. The addition of some very nice members. And yeah, we're having a good time analyzing animal venoms at different levels and trying to merge not only the compositional analysis, but mainly like having a strong focus also on the functional part. So in line with what Margareta said, I'm also about to present a bit what have been the results of my short-term scientific mission granted by EUVEN. And it started as something that I obtained right after submitting my PhD thesis, you know, when you are pretty much in the limbo of, oh my God, what am I going to do with my life before I actually defend my thesis? So in those stressful months, it seemed like a very good idea to try to get a bit more experience in venomics than what would have been available at the University of Porto. So EUVEN just was perfect in offering this opportunity to be a bit more international. And so it granted me the first contact ever with the animal venomics group of Tim Ludeca here in Germany. And the idea of the project was like, yeah, let's just, you know, compare the venoms of blunt-nosed vipers, genus Macrovipera, which are these quite amazing vipers distributed from Greece all the way to Pakistan and northern Kashmir, actually. So yeah, it was like, great opportunity. Let's see how this leads. Let's see how this goes, and so on. You know, good expectations, of course, but really just more of an explorative thing. And to be fair, this turned out to be quite the change in my career, because the output has been, personally, I really found it quite amazing. It has been quite shocking. EUVEN basically allowed me to stay for 3 months at the Animal Venomics group. And as you can see, there is a bunch of very nice and friendly people all working with venoms. So it's a group of maniacs. And to be honest, I think I fit quite well with them. So I had a great time. And the short-term scientific mission led to the publication of the venomics of the Milos viper, again, a blunt-nosed viper. First, for this species, so quite interesting, and with a lot of actually exochemistry assays, biological activity assays across the genus Macrovipera, which actually became my main focus, to the point that thanks to the results of the EUVEN STSM, together with Tim and Wolfgang Wüster, we applied for a grant by the DFG, which is like the German Research Institute, for a postdoc position, and actually we got it. So yeah, thank you very much, EUVEN, with your short-term scientific mission. Basically, you granted me a job for at least the next 2 years, which is great, and the opportunity to develop my own project on Macrovipera, which is called EUVEN and is currently in development. So yeah, great stuff. Thank you very much. Really, really, really appreciate it. Really grateful for it. And this is a resume of the STSM, but I think that now I can go through the real topic of the talk, which is the analysis, the venomics analysis of the sharp-nosed viper venom. Starting from a bit of background, what is snake venom? It has already been said by Margareta. It can be considered an evolutionary innovation of the advanced snakes, the so-called Chenophidia, mainly composed by proteins and peptides, although other biological active compounds are present. It can target different systems, of course, because we know there are neurotoxic venoms, hemotoxic venoms, etc. It is complex and highly variable, as it's already been said. It can vary depending on different factors, but some of the main drivers seem to be predation and defense. Predation seems to be the leading driver of snake venom evolution and variation, while defense has been confirmed just for a bunch of species, but very charismatic ones, namely spitting elapids, the cobras, and wrinkles. Snakebite problem, of course. So when you talk about snake venom and it being used for defense, one of the main examples that we can that comes to our mind is, of course, the snakebite problem. It is a neglected tropical disease, recognized as such officially by the WHO World Health Organization only in 2017. The hotspots of snakebite are in the global south, as we can see here. The darker colors are where the incidence of snakebite is the highest. Yearly are registered officially approximately 5.4 million of cases around the world, cases of snakebite. And of course, it has huge impact on health and economy because it can lead to long-term sequelae other than death. And the WHO aims to halve it by 2030. So there is quite some interest put into this topic. Talking about snakebite, we have to talk about the medically relevant snakes, which are basically the snakes that are recognized as potentially causing disabilities or death with their bite by the WHO. And they can be largely divided into front-fanged snakes and non-front-fanged snakes. So in front-fanged snakes, here we can see the venom delivery system is characterized by the fang in red, venom gland in gray, and there is a compressor muscle right above the gland or in its close proximity, which of course squeezes the gland and lets the venom get out through the venom gut into the fang and into the bitten subject. The main difference with non-front-fanged snakes is the lack of the compressor muscle, although it has been seen that some non-front-fanged snakes do have some kind of muscle around the gland. And all in all, we can say that front-fanged snakes are the infamous Viperidae, so vipers, Elapidae, meaning cobras, king cobras, coral snakes, and so on, and Atractospiridae, which are commonly known as the burrowing asps or stiletto snakes. And pretty much non-front-fanged snakes can be recognized as everything that is not front-fanged. So it includes all the so-called Colubridae, there are some Ophidae in it, and other taxa. Through bibliographic analysis, it has been seen that Viperidae and Elapidae are consistently more studied than other snake groups. And particularly with the pit vipers, the subfamily Crotalinae being the most investigated group. And what has been seen is that the harmful potential of the snakes seem to play a strong influence on its popularity in snake venom studies, as it would be expected in the context of the problem of snakebite. And talking about pit vipers and very, very dangerous snakes, today I will talk about the Denankistron acutus, which is this nice fellow here, an Asian pit viper, so belonging to the subfamily Crotalinae, Here, the distribution is taken from the World Health Organization Snakebite Information and Data Platform, so it's quite updated, I think, to 2021. It is a fixed, large snake, reaching approximately 150 centimeters in total length. As we can see, it's quite heavy built, and it is known as sharp-nosed viper for this characteristical pointed snout that it has. Also, it is known as the 100 pacer because local people think that after a bite from the snake, you can survive only up to 100 steps before succumbing to your death. It is the object of a lot of interest, also like not only scientific, but also, I would say, ethnological, ethnopathology, I think is the term. Because it has been considered also as a protector, like some kind of divine animal by several people in Taiwan, for example, because of the properties and the potency of its venom. And it has been analyzed through different high-throughput technologies. For example, genomes of the species are available, transcriptomes have been available, proteomes, venom proteomes are available. Still, although so important and so famous and so infamous, these technologies, this body of knowledge has rarely been combined. And of course, this snake is characterized as most viperids by coagulopathic, serotoxic, and hemorrhage-inducing venom. And it is considered of the highest medical relevance by WHO across most of its range. Now a little bit of a disclaimer, because I'm going to show some pictures that are not particularly nice following the Nankistan acutus animation, which, although not very nice, I think are needed to understand exactly what is the relevance of the snake. So here we got a case of a guy in Taiwan that at night, after some beers with his friends, just went home, you know, like the calmest night that you can have with your buddies. You cross through a field, little garden, you get bitten on the leg, as we can see on the first figure from the right, and the day after, he didn't wake up. The effects were strongly hemorrhagic. As we can see in the first picture, you can see these hemorrhagic bulle, basically blisters filled with blood, hematoma, bruises extending everywhere, and lots of necrotic tissues, as we can see in the picture in the middle. Additionally, another case of snakebite on the hand delivered by the Nankistan acutus caused a very big aneurysma in the veins in the wrist that caused compartment syndrome and eventually necrosis of the area, which amputation of the limb. And in tests done on mice, it has been clearly seen that the venom is potently hemorrhagic, causing a lot of hemorrhage in internal organs, particularly affecting the lungs and the liver. So yeah, definitely this snake is 100% medically relevant. If you get bitten by this species, you should immediately go to the hospital, and it is really no joke. Given its relevance, we thought that it would absolutely be necessary to delve a bit more into the composition and the activities of the species venom. Therefore, we aim to contribute to the current knowledge of the venom of the sharp-nosed viper by presenting the first proteogenomics-based catalog of the protein composition of the venom of this species, provide a comprehensive picture of the biological activity of the venom of sharp-nosed viper through extensive activity profiling, ultimately offering a valuable foundation for understanding and possibly treating bites by the species. To this end, we applied a proteogenomics-based approach. So first of all, the first step is, of course, the venom collection from the Nankistan acutus and the lyophilization. With the lyophilized venom, we first went through venom profiling through standard means, I would say, reverse phase product liquid chromatography and STS page. Then we got the proteogenomics approach, which was a shotgun base. So we basically developed shotgun proteomics and eventually combined the information that we got to the shotgun analysis with genome-based identification of the venom components. Of course, having a genome available allowed us for species-specific identification, therefore highly enhancing the identification of the compounds and avoiding the problem that comes with using public available databases. And last but not least, we applied a lot of bioactivity assays on different targets typical of viperidum venomation Like we analyze protease activity, PLA2, phospholipase A2 activity, hemostasis altering activity, cell viability, and also second messengers. And we got an idea of how the venom of this species can affect these targets. In terms of venom composition, our proteogenomics-based analysis revealed the presence of 117 proteins identified in the venom sharp-nosed viper. Ultimately, we found out that several of them were actually physiological proteins, and therefore we were able to ultimately select 45 bona fide venom components and categorize them into 18 distinct protein groups. We found that the compositional pattern of this species follows the general compositional pattern described for viparid venoms, with a lot of SVMP, metalloproteases, phospholipases A2, serine proteases, and CTL, C-type lectins. And these main components are in line with the hemorrhagic, coagulotoxic, and serotoxic effects described for this viper. Concerning the biological activity, we tested different activities like protease activity, PLA2, the one said before. We saw a very strong, very positive concentration-dependent effect, with the highest concentrations having, of course, the higher effects. Still, the effects of the protease activity and the phospholipase A2 activity were quite remarkable. As we can see, for example, in the PLA2 assay, at the highest concentration, we can get an effect that is comparable to the positive control, which is quite something. We found weak hemolytic activity, weak effects on factor Xa and thrombin activity, and no plasmin activity was detected at all, which was quite interesting. And considering the serotoxicity of the species, we tested the venom on different cell lines, which were like epithelial-like and epithelial lung and kidney cells, neuroblastoma cells, macrophages, and so on. And we found that there were definitely some very, very strong effects, as you can see here, on the cell viability of the kidney cells, MDCK2, and the HEK293T, and on the lung cells, A549. While we got weaker effects on the neuroblastoma cells, SHSY5I, and no activity was detected on the other cell lines. These are pictures of the actual cytotoxicity assays developed on the lung cells, A549, and on the kidney cells, MDCK2. And as we can see, like at the highest concentration, which is 25 micrograms per milliliter, we really have a total clean background, which is insane. Like on the top, you can find some, little black bulbs, and you might think that those are cells because yes, they are, but they are all dead. Those are clusters of dead cells. It is quite impressive to me to see that at the highest concentration, the venom of the Rankistron acutus really does a tabula rasa of all the cells, even more than ionomycin, which is actually the positive control that we were using. So that was really, really quite impressive. Therefore, we got a nice set of results to discuss. And we provided the first proteogenomics-based investigation of the Rankistron acutus venom, which constitutes the most accurate venom protein catalog for this species produced to date. Because as I said, yes, you can use the online databases, the freely available ones like UniProt and so on. But of course, there will be some problems due to the lack of curation, general lack of curation of these datasets, of these databases, redundancy, and so on. So having a species-specific database for comparison is definitely needed for proteomics analysis. We found that the venom of the species is relatively simple, with the major components that align with the typical symptoms of the Rankistron acutus envenoming, and to be fair, with the general symptoms also seen in the venoming by other viperids, so typically coagulotoxic, hemorrhagic, and cytotoxic. We found high levels of protease and PLA2 activity, which are concordant with potent serotoxic activity. In fact, proteases and PLA2 can exert a variety of effects, but among them, tissue damaging effects are probably the most known ones. We found a high affinity for the epithelial-like kidney cells and epithelial lung cells in the serotoxicity assays, which is actually consistent with direct impairment of kidneys and lungs reported for envenomation cases by sharp-nosed viper. In fact, some of the most common symptoms, as we saw in the pictures, are hemorrhage of the lungs, for example, and also renal failure and renal impairment. And also, when I showed the analysis of the assays on the thrombin, plasmin, factor Xa, which we saw that the bars were very low, therefore there was very little activity on these coagulation-related assays, it appears to be in line with the mainly anticoagulant venom profile. And this is something that I would be actually very interested in testing next, because it has been reported in previous analysis that the venom of the Nagistran acutus is a anticoagulant or pseudo-anticoagulant, like basically when you get bitten by this species, your blood stops clotting. And to be fair, if you find very low amounts of factor Xa and thrombin, I would tend to believe that, because of course those help the coagulation. And so yeah, that seems to be in line with what has been reported previously. And I'm also very happy to say that the results of these studies have actually been published last week. Yeah, I think last week on International Journal of Biological Macromolecules. So yeah, it has been a very nice journey. It has been a very nice trip. This work started actually last year already during the STSM with the preliminary analysis on it. So around December or so, and I'm very happy to see it out now that it is August. And yeah, it was really, really great stuff. I need to acknowledge and thank an insane amount of people, the institution that hosted me, of course, my collaborators for the article, the people that actually gave me these amazing pictures that were used both for the presentation and for the article. So yeah, thank you very much. Thank you, EUVEN, for providing all this, for making all this possible. And yeah, thank you very much for your attention.
Thank you, Ignazio. Wonderful talk. What a horrible animal. And you're giving Sir David Worrell a run for his money for most disturbing clinical images. I've seen two of his talks in recent weeks, but yours were grosser.
Sorry about that.
Any questions or comments? Maybe I'll give myself the first dip. Is there any reason to expect why this animal seems to have such a strongly destructive venom based on its natural history, prey it eats or something, or is it a bit of a surprise?
So it is reported to be a typical biparid landmine, an ambush predator that stays on the on the soil, on the bottom of the forest, just waiting for something to pass by. And when you are an ambush predator, you really want to be sure that you kill whatever you bite, because otherwise you will have to wait for another pretty long time, before something passes by. And definitely it has been reported to eat mammalian prey, endotherm prey, like big rats, big rodents, and so on. So I think it kind of makes sense that it has a venom that strongly attacks mammalian cells, which are in the end the ones that we tested. I think it kind of makes sense if you consider the diet of this animal, and again the need to be particularly strong.
Thanks. Lorenzo, you raised your hand first.
Oh yeah, but I'm happy to let Dr. Yanis ask first, since I already asked the question before.
All right, Yanis, do you want to go ahead?
Yeah, okay. Ignazio, good afternoon.
Yanis, nice to hear from you.
Okay. Did you notice any ultra short peptides as the other times in macroviperas?
Sorry? Ultra?
Ultra short peptides, like in macroviperas.
Small peptides in the Ankistrodon.
Yes.
I can have a look actually right now. Short stuff. We found some. We didn't find inhibitors. If that's what you're talking about. Yeah, I don't think.
That's why.
Yeah. You're right. Yes. Yeah, it was something that we were discussing with Michael. So yeah, we didn't seem to find inhibitors as far as I remember. I don't think we found these very short ones. Yeah.
Okay. Okay. Thank you.
No problem.
Do you mean protease inhibitors, or what kind of inhibitors?
Yeah. You can get usually you find also in the chromatograms, you usually see them in biparid venoms, these short metalloprotease inhibitors.
Yeah.
But yeah, as far as I remember, we did not find them in these species.
Any other comments or questions? Or if not for Ignazio,
I think there was Lorenzo still that wanted to ask something, or maybe change his mind. I don't know.
No, if there's still time, yeah, definitely, definitely do have a question. First off, Dr. Jenner, I have to disagree. I think the Inankistron acutus is actually one of the most beautiful vipers on the planet.
Tell him, Lorenzo. Tell him. Yes.
But aside from that, yeah, I was at the World Congress of Herpetology last month. There was a poster about maternal care being documented in this species, and that absolutely blew my mind. And it also led me to think, again, as a follow-up to what Professor Jenner was saying about what we know about the ecology of venom in the Inankistron, because it's like you said, it's an absolute landmine, and you show its distribution before. It goes from Western China all the way to Taiwan, passing like mountain ranges, rivers, forests, and all that. So do we know anything about venom variation or any other sort of factors that could influence venom phenotype in this species?
Yes, we do, because actually, funnily enough, I think this year some articles have been published on venom variation in this species. It has not been analyzed through proteomics, as far as I remember, but just through HPLC profiles, and there was something. There was some variation, which is expected, right? Because in the end, venom variation occurs pretty much at every level. And also remember that I think in 2023, so last year, there was an article published on the differences in the effectiveness, yes, I think that is the right term in English, of antivenom on different population of the Inankistron acutus. Also, there is a phylogeographic analysis on, like, possibly there are two major clades, you know, divided by, I don't remember exactly which kind of barrier. I think must be a big river or something in its Chinese distribution. So possibly there is also quite some genetic variation within the Inankistron acutus. And although it is currently recognized as one species, I wouldn't be surprised if in the future, you know, someone pops up and says, no, actually, you know, what we've been considering one species is more than one. And yeah, there is definitely variation in the venom of this species.
Yeah, so in short, I should have done my homework before this. Thanks, man.
No, just read our article and cite us.
Congratulations with your article.
Thank you.
There's a question in the chat. A person with a very weird name, user's iPhone, wants to know, what is the most dominant toxin in this viper, and do you think antivenom developed for other viper species can be useful in treating victims of this snake?
So the most dominant toxin, from what we saw, maybe I should have mentioned this, like with our proteogenomics analysis, we didn't only get information on the diversity of the components within the venom, but also about the abundance. So actually the pie chart that I showed shows some relative abundances. And therefore, as we saw it, the most abundant toxin family seems to be CTL, C-type lectins, and C-type lectin-related proteins, so SNAC-lex, how you want to call them. This is a toxin taxonomy, I would say. So yeah, they have mostly coagulopathic effects, as far as I remember. And do you think antivenom developed against other vipers can be useful in treating victims of these acutus? You should really check, like, to answer this question, I think, first of all, testing, of course, because there's so much variation within toxin families that I would lie. Like, I can go for an educated guess and maybe assume that some closely related pit viper from Asia might possibly produce some similar toxins, but I wouldn't really know, to be fair.
I know, Tim, that you have raised your hand. However, I also know that you are meters away from Ignazio, and in the interest of time, and also to get you off the hook, Ignazio, I want to thank you again for your lovely talk. And